Have you ever really thought about how a fleeting experience, like a conversation you had yesterday or the route to a new coffee shop, actually gets physically locked into your brain? I mean, how does something you can't even touch become a real biological thing?
Right, let's get into it. We are going to break down one of the most amazing processes in all of biology. How your brain literally transforms a moment in time into a physical memory trace, something scientists call an engram. So to figure out what's happening on the inside of the brain, scientists first have to see learning in action on the outside. And that means designing some pretty clever tests that can show us, without a doubt, whether an animal has actually formed a memory. Okay, so our first stop is the behavioral lab. Because you see, before we can even begin to look for a memory inside the brain, we've got to prove that one has been made in the first place. And for that, you need a task and you need data. This right here is a classic, the Morris water maze. It's a fantastic test for spatial learning. So here's the setup. A mouse gets placed in a pool of cloudy water. Somewhere in that pool, there's a small, hidden platform where it can rest. Now to find it, the mouse can't just sniff it out. It has to look around at the different cues in the room and build a mental map of its surroundings. And what you see is, over a few tries, a mouse that's learning will find that platform way faster. But here's the real kicker. This is the proof. In what's called a probe trial, the scientists take the platform out completely. Now look at the chart. A mouse that remembers where the platform is supposed to be, that's our control group on the left. It spends a huge amount of its time searching in that one correct spot. But an animal with impaired memory, it just swims around randomly. That data right there is how you can actually measure and quantify a memory. It's brilliant.
All right, so we've confirmed the memory is real. So the next obvious question is, where? Where in that big, complicated brain is this memory actually being made? It's time to zoom in from the behavior to the actual brain anatomy.
And you're not going to believe this, but the story of where memories are formed actually starts with this little guy. Yeah. Early anatomists were looking at the structure deep inside the brain, and they thought, hey, that kind of looks like a seahorse. Or in Greek, hippocampus. And honestly, you can see why they thought that. On the left is a real cross-section of the hippocampus, and the resemblance is just uncanny. This seahorse-shaped structure is absolutely essential for forming new memories about facts and places, exactly the kind of memory we just saw being tested in the water maze. You can think of it as a master hub, tying all the different parts of an experience together. So we know where the memory is being processed, but that's just the neighborhood. We need to find the specific address. So we're zooming in again, going from the hippocampus as a whole down to the microscopic connections between the neurons. This, right here, is where the magic really happens. And it all comes down to one core concept: synaptic plasticity. It's a simple idea, but it's incredibly powerful. It just means that the connections between your brain cells, your synapses, they're not set in stone. They can get stronger or they can get weaker, all based on how active they are. This is literally your brain physically changing in response to experience. There's a famous saying in neuroscience that just nails this idea perfectly. Synapses that fire together, wire together. Basically, if one brain cell repeatedly helps another one fire, the connection between them gets stronger, faster, and more efficient. It's like they form a special link. This is learning at its most basic, cellular level. The main way this happens is through a process called long-term potentiation, or LTP. Just think of it like this. When one neuron sends a really intense, high-frequency burst of signals to another, it's not just talking, it's shouting. This causes a huge release of a neurotransmitter called glutamate. The receiving neuron gets super activated, and that kicks off a whole bunch of changes that physically strengthen that connection. It's like upgrading a tiny footpath into a multi-lane highway. And that potentiation, that strengthening, can last for a really long time. Okay, time for our final zoom in. We're going all the way down. What are those physical changes? What is the actual molecular machinery that takes a temporary electrical buzz and turns it into a permanent physical memory? Now, I know this looks really complicated, but just stick with me. It's basically a chain reaction, like dominoes falling. That big shout from the last slide lets a flood of calcium ions rush into the cell. Calcium is messenger number one. It wakes up these enzymes called kinases, which are kind of like little project managers. Their job is to run around and activate other proteins. This whole relay race keeps going on until the final message gets to the cell's main office, the nucleus, and activates a master switch molecule called Krebs. And understanding this pathway is actually the key to seeing the difference between short-term and long-term memory.
See, short-term memory is kind of like a quick software fix. It just tweaks proteins that are already hanging around at the synapse. But for a memory to become long-term, you need CREB. CREB is what's called a transcription factor, which basically means it can march into the nucleus, turn on specific genes from your DNA, and order the cell to build entirely new proteins and structures. You're not just tweaking the synapse anymore. You are fundamentally rebuilding it to be stronger and last longer.
So we've gone from a mouse swimming in a pool all the way down to a single molecule inside a single cell. Now let's zoom back out and ask a big question. Is this whole system something special? Something that only advanced brains like ours have?
And the answer is a resounding no. It's actually incredible. When scientists look across the animal kingdom, they find these exact same molecular tools over and over again. That rush of calcium, the activation of kinases, and even that master regulator CREB, it's the same basic toolkit used for memory formation in all sorts of different creatures.
And it's a profound biological truth. The basic machinery for changing a connection between neurons based on experience is ancient. The core process that lets a simple sea slug learn is the very same process happening in your hippocampus at this very moment. Evolution found a great idea, and it just stuck with it for hundreds of millions of years.
So there you have it. From a behavior you can measure, to a structure you can see, to a connection that gets stronger, all the way down to the molecules that rebuild it. That is how an experience becomes a physical memory. It's the literal trace of your past written into the very wiring of your brain. Which kind of leaves you with one last big thought. If you are the sum of your experiences, and those experiences are physically stored in these connections, who would you be without them?